Hi, my name is Edenir Pereira Filho, and I am professor at the Federal University of São Carlos, São Paulo State, Brazil. Here we can find my contacts, my email, my YouTube channel, LinkedIn, Instagram, and also Facebook, and the homepage of my research group. Today, I will talk about a very special equipment, a J200 instrument from Applied Spectra. The title of this presentation is the J200 Instrument Saga, 10 Years of Analytical Chemistry Development Using LIBS. Okay? Then, what is LIBS? Laser-Induced Breakdown Spectroscopy. The first publication about LIBS was in 60 by Branch and Cross, and the first analytical application was published in 1963. And nowadays, the importance and the number of applications using LIBS in several fields like industry is increasing. But how LIBS works? For example, we have analytes, for instance, in chromium, lead, cadmium, and when we focus at a plasma, a high energy plasma in this type of sample, we can have vaporization, atomization, and ionization of the constituents. These constituents can present also an excitation of atoms and ions. And then, after, we have an emission of these elements. And of course, this emission is characteristic from each element, and then we can perform quantitative or qualitative analysis. But how LIBS works? LIBS employs a high energy laser pulse from 10 to hundreds of millijoules and a focused lens to generate plasma that vaporizes a small amount of samples, microgram, nanogram of samples. Then we have an excitation. During this excitation, the analytes in the plasma dissociates into various ionic and atomic species. As the plasma cools, the excited ions and atoms emit optical radiation. How is the mechanism of LIBS? Using LIBS, we need to control the time. For instance, in time zero, we perform the laser pulse in the surface of the sample. Then we have a continuum, a radiation named continuum. This radiation, we need this radiation decrease, and this time of for the radiation decrease, the intensity is named delay time. After that, we have ionic, atomic, and also molecular emission. And in this area here, in this time, we can perform determinations of the analytes. Okay, let's continue. What's the advantage of LIBS? LIBS is a versatile for some solid samples, gases, and also liquids. Little or no sample preparation is necessary. You can analyze direct solid samples. Then, as the time for sample preparation is reduced, we have few opportunities for contamination. We need small amount of samples. These samples can be conductive, can be not non-conductive. We can analyze very hard samples like glasses, metals, alloys, ceramics. We can use LIPS in hazardous environments and also in long distance. We can perform, for example, analysis of microregions, for instance, and we can perform also simultaneous multi-elemental analysis. LIPS has potential to direct analysis of aerosols, and the analysis is very fast. In few seconds, you can obtain several spectra. Motivation. What's the motivation of the use of LIPS? During the analysis, we need, in the majority of the analytical techniques, we need to prepare the sample. We need to dissolve the sample in a liquid medium in order to analyze the sample in an ICP or ES, for example. But using LIPS, we can perform direct soil analysis. Then the best way to prepare a sample is not to prepare a sample. But as we are not preparing the sample, we can face several problems like spectral and matrix interferences. We have micro heterogeneity of the sample, how to select a calibration strategy for quantitative analysis, how to select appropriate standards for quantitative analysis, how to prepare the standards. We can use certified reference materials, we can use reference samples, we can use some samples with known concentrations. Okay, how to minimize these limitations? We can use chemometrics. Chemometrics is a science to plan or select optimal experimental conditions and to provide maximum chemical information with data analysis. And using chemometrics, we can perform optimization, normalization, or standardization of the data, multivariate models, univariate models, new and rediscovered calibration strategies can be using LIBS, and also you can perform classification models in order to perform qualitative analysis. In 2021, we published this ebook, and this ebook is totally free, and it was published in two languages, English and also in Portuguese. In this link here, this link is also in the video description. You can freely download this ebook. The name of this ebook is Laser-Induced Breakdown Spectroscopy, LIBS, Applications and Calibration Strategies. And now I will present some results obtained using the J200 equipment from applied spectroscopy, and these results are also described in this ebook. Sample preparation for LIBS analysis. The first topic. We can perform direct analysis of solid samples. In this way, we have minimal or no pretreatment of the samples. We can have high analytical throughput using analysis, during the analysis, reduce the risks of analyte losses, reduce use of hazardous reagents like acids, and of course, reducing the generation of waste. A small amount of sample is required. We can perform also spatial distribution of the analyte in the sample surface. How to prepare solid samples? If the sample is heterogeneous, we need a grinding and homogenization step. We can analyze biological, environmental, geological, food, and e-waste samples. If the samples are homogeneous, like glasses, alloys, and polymers, we can introduce the sample directly into the instrument. In some cases, the sample is a powder. Then we need to prepare a pellet in order to present this for the analysis. In some cases, the sample is not totally hard, and then we need to mix the sample with a binder like polyvinyl alcohol, like cellulose microcrystalline, and then we can perform the pressing process in order to obtain the pellet. Sample preparation for liquids and suspensions. We can immobilize the sample using, for example, a polymer like polyvinyl alcohol, or we can freeze, we can perform electrodeposition of the analyte in a substrate, we can encapsulate the sample with PVA, polyvinyl alcohol, or paraffin, freeze. We can also prepare a liquid jet and focus the laser in this jet. We can adsorb the liquid sample in carbon, for example, in wood, in bamboo, or we can precipitate or perform electrodeposition of the liquid sample. Optimization. In LIBS, we have several variables in order to optimize, like the laser pulse energy, the spot size, the delay time. In this optimization, we must use, for instance, design of experiments and calculate some statistical metrics like ANOVA table. We can use response surface methodology. My research group and I prepared a YouTube channel. The link of this YouTube channel is here and is also in the description of the video, with several playlists about design of experiments, about PCA, principal component analysis, and other topics. Data processing. The main goal of the data processing is reduce signal fluctuation, perform variable selection. We can use some mathematical transformation of the raw data and also improve figures of merit like limits of detection and quantification. One of the data processing that we can perform is normalizations. In my research group, during the last 10 years, we developed several normalization procedures in order to minimize the laser pulse fluctuation, minimize the micro heterogeneity of the sample. And these normalizations are, for instance, normalization by the norm, by the area, by the maximum. And we are using also carbon as internal standard. And the main goal is data without normalization. It's like that. Data with normalization. We are performing something like that. Okay. In the normalizations, we can also observe outliers. We can remove these outliers and calculate, for instance, peak area and also peak height, and also signal to background ratio of the selected wavelength, selected emission line. We perform also a baseline offset correction. For instance, the sample has a spectrum like that. And after BOC, we are performing a baseline offset correction of the data. Univariate calibration, some examples of univariate calibration. The first strategy employed in LIBS was matrix matching calibration. We can use, for instance, certified reference materials or samples that present reference values. We can add in these standards and samples an internal standard for instance, yttrium, scandium, or use an element that's naturally present in the sample, for instance, carbon and other elements. There is also standard addition, and in the majority of the applications, we can mix the sample and standards with microcellulose in order to prepare the standards, in order to prepare the pellets. One of the first non-traditional calibration standards employed in LIBS was MEC, multi-energy calibration. MEC was first proposed for ICP-OES, and the main characteristic of MEC is to prepare two pellets of sample. The first one presents a sample and a standard, and the second one presents a sample and a diluent. And then we analyze the sample in different wavelengths. For instance, iron, chromium, these elements present several wavelengths, several emission lines, and then we can build a linear model, and the concentration of the analyte sample is the slope of this linear model times the concentration of the analyte in the standard that is known, divided by 1 minus the slope. Using MEC, we can perform also the identification of spectral interference. In the case of a wavelength that is not in the linear model, for example, this wavelength here is far from the model, this is probably interferent. And then we can remove this wavelength and calculate the model again. The first application, as I told before, was in ICP-OES, and now we are applying this strategy in LIBS with very good results. In LIBS, we applied this in mineral supplements and also in electric waste, electric and electronic equipment, in also ores. Other strategy is two-point calibration transfer. In two-point calibration transfer, we prepare two pellets of sample. And we prepare a pellet for sample and for standard, and apply several pulses in both. The first set of that data, for instance, has around 100 pulses, 100 accumulated spectra, and the second one, 300 accumulated spectra. Of course, the second set of data present a higher signal, and we have here a linear model, and calculating the slope of the linear model for sample divided by the slope of the linear model of the standard times the concentration of the analyte in the standard that is known, we can calculate the concentration of the analyte in the sample. We apply this strategy for hard drive magnets from electric waste equipment, and we determined earth elements. Multivariate calibration. In multivariate calibration, we can improve the precision and robustness of the method. It's possible to handle interference. We can use several emission lines from the same analyte in order to calculate linear models, and we can use, for instance, multiple linear regression, principal component regression, and partial least square. In other words, we can combine a multivariate technique with mathematics and statistics and obtain a powerful combination. Some examples. The first one is PLS, partial least squares. PLS is the most used chemometric technique for multivariate calibration. We need to perform a validation data set. We can separate the data in calibration and the second set for the validation. In the first one, we calculate a model and test this model in the validation. It is necessary reference samples with reference concentrations. Interferences can be handled if present in the calibration data set. And we apply this strategy in polymer analysis, for example. A new strategy that we applied in LIBS was unfolded partial least square with residual bilinearization. What's the main goal of this? We can use, for instance, mixture of salts. We can prepare calibration data set with salts. For example, we need to determine magnesium. We can mix magnesium chloride, for example. The other analyte is sodium. You can prepare a mixture of salts in order to prepare a standard. And the sample can be a different sample, for example, a sample of supplementary food, for instance. And we have a residual bilinearization at the end in order to compensate the differences of matrices from the standards and from the samples. Another example is PARAFAC, parallel factor analysis. And using this strategy, it's possible to identify and remove interference. It is necessary a data array. This data array can be emission lines, different delay time, and ablated position and depth in the sample. Another application, another strategy for calibration is data fusion. We can combine, for example, LIBS data with X-ray, for instance, with ICP, MS, in order to obtain better calibration models. We apply this strategy in electronic ways that I will present in the next slides. Some applications. Now, the J200. This is the J200 equipment from Applied Spectra. This instrument is in our laboratory since 2012, and we perform several types of analysis in this equipment. This little door here, we can open and introduce a sample from the size, for example, of a mobile phone, a cell phone. Let's see some applications. The first set of applications that I will present is for food analysis. We determine calcium, potassium, magnesium, and phosphorus in cocoa and also in frost samples. There are other applications for beef authentication using LIBS. Here we have some examples, some results for cocoa beans, the results for calcium and the results for potassium. We tested several univariate calibration strategies like single sample calibration, 2-point calibration transfer, and the trueness, the accuracy of the results were around 80 to 120% for calcium and also for potassium. The errors, for example, the results for calcium, the concentration of calcium in cocoa beans are from 0.06 to 0.15%. And the errors for calcium are around are lower than 0.03. The same we can see for other elements like potassium and also magnesium. As you can see, the errors are lower than the concentration of the analytes. Applications for waste electrical and electronic equipments. This type of sample is very complex because you have several types of elements. Uh, borium, iron, hard earth elements, antimony, lead, aluminium, copper, iron, lead, and also indium in the LCD screens. And the antimony is used as flame retardant in this type of sample. We have here an example with hard drive magnets. This is the actuator magnet. And we performed, as you can see in this square, several laser pulses were performed in this area. There is a magnification here, and the analyzed area is around 1.52 by 2 millimeters. We can see, for example, this part here is rich in nickel, and this other part here has a high concentration of neodymium. You can see here that the film of the magnet was removed, and we analyze part of the substrate and part of the film. The film is composed by nickel, and the magnetic is composed by neodymium. Some results for hard earth elements, like dysprosium, gadolinium, neodymium, praseodymium, samarium, and terbium. The results for relative standard deviation varied from 2 up to 28% using two-point calibration transfer. And the results for trueness varied from, for example, using two-point calibration transfer from 8% up to 129%. The problem we found was about terbium. The results were not so good because the concentration of terbium are very low in this type of sample, the hard drive magnets. Another application was copper determination in printed circuit boards, PCBs, printed circuit boards, and using Parafac, we observed a presence of an interferent here. You can see here an interferent, and using Parafac, we removed this interferent and later performed multivariate calibration using PLS, for example. This is another application using combining Parafac and LIBS. Another combination of LIBS and principal component analysis. We have here the optical image of two printed circuit boards, the front side and the back side, and using LIBS, we can perform a heat map in order to see the composition of the samples. For example, in the front, in this area, here in this area that I am drawing this red line, is rich in gold, potassium, and nickel. This area here also. And this area that is in yellow, is rich in barium, the back side here, this area presents high score values, and this area is rich in nickel. The blue part is rich in copper, aluminum, barium, magnesium, silicon. These are some results that we found for electronic waste. Data fusion. Then, our main goal here was to determine copper in this type of sample in order to perform a recycling process after. In x-axis, we have the reference copper concentration, and in the y-axis, the predicted concentration. We selected an emission line of copper for LIBS, and the Turner's values, you can see, varied from 17 up to 200%, or not so good. Using X-ray fluorescence, the Turner's values were from 50% up to 140. Then we combined both results, both signals, the copper from LIBS and the copper from X-ray fluorescence, and the results were much better. The same samples, the Turner's varied from 8 to 150, 20%. And we have here some results, some comparisons, and you can see, for example, the results for data fusion. We performed here a normalization using Euclidean norm. We use the area for copper signal, and the Turner's values varied from 81 to 119%. Some results for sugarcane. We use LIBS in order to analyze the fiber content in sugarcane. And also, we analyze fertilizers in order to determine phosphorus, for example, and also some contaminants in fertilizers. Some fertilizers were liquid, and we encapsulated the samples using polyvinyl alcohol. I am presenting here some results for calcium in fertilizers, and we performed in the first case a matrix method calibration using reference materials, a matrix method calibration using reference samples, and multi-energy calibration. We can see that the recovery, the true values, were very good for multi-energy calibration when compared to the other strategies. And we test also internal standard in order to improve the calibration process. We test also another strategy, that is one-point calibration standard addition, one-point gravimetric standard addition, and the recoveries varied from 82 up to 115%. We use LIBS also for archaeological measurements. In this picture here, we have a coprolite. A coprolite is this sample here. Coprolite is a feces of dinosaurs that is in a rock substrate. And then we performed several laser pulses in several points, and using PCA, we are able to identify, qualitatively speaking, the composition of the rock and the composition of the coprolite, and see how the animal, what type of food this animal consumed. Considerations, conclusions, and perspectives. Conclusions and perspectives. Using LIBS, we can open new frontiers For example, LIBS now is using in space exploration. We can perform data mining because we have several hundreds, thousands of data. We can solve complex problems as in underwater measurements. We can use our imagination and also cooperation among different areas like chemistry, physics, and mathematics in order to develop new methods, new calibration strategies that are useful for industry, that are useful for metals determination, for minerals analysis. Our research team. The research team was composed by the J200, that is here, the instrument, and also for former students that contributed in these results. And thank you very much for your kind attention, and I am open to answer some questions, to discuss some results, and also for collaborations. Thank you very much for your kind attention.